Hi, my name is Dr. Robert Groysman from covidinstitute.org, and I'm going to be talking today about chromosomes, genes, base pairs, nucleotides, DNA, RNA, and amino acids. Why am I mentioning this in the context of long COVID? Well, it's simple. The virus can damage some of your cell's DNA. It's promoting oxidative stress in your cells during the infection and even afterwards. So, why is it important to understand what genes are and how they work? What is their purpose? Every single living organism, whether it's bacteria, plants, or animals, has a different number of chromosomes and different genes. Now, what's interesting is that the chromosome number does not correlate with how advanced an organism is. Fruit flies, for instance, have 8 chromosomes. Rice plant has 24. A dog has 78. A giraffe has 30. Baker's yeast has 32. Brown bear, for instance, has 74. And us as humans, we have 46 chromosomes, 23 pairs. We get 23 from mom and 23 from dad. So again, why is this important to know? Why is it important to know about DNA during COVID and long COVID? It's because there's DNA damage occurring. There's increased oxidative stress and damage. There's decreased absorption of vitamin D, and these are important in DNA repair. Increased immune response. DNA damage has been positively correlated to interleukin 6 concentration and negatively correlated with platelet counts. This is just something of a marker. High level of CRP or C-reactive protein were reported when there's oxidative damage occurring in DNA. Oxidative DNA damage is also associated with chronic diseases and certain types of cancer. A single human chromosome stretched out would be about 2 inches long. Now think about this. This is something that fits inside of a cell, something that's microscopic. If you were to take all 46 chromosomes and stretch them out end to end, it would measure 6.5 feet. That's from a single cell, laid end to end, 6.5 feet. It's taller than most people. We have about 3 billion base pairs in our genome. Each chromosome has thousands of genes on it, and we have an estimated of about 100,000 genes. About 6,000 is what we know what they do. The rest we're still not sure about. There's a lot of unused genes and DNA material and non-coding areas inside our chromosomes. Most of our genes reside in about 2.5% of our entire genome, so a very small part. DNA in our cells is wound and compressed in several ways. Think of it like a zip file. When we look at our chromosomes, it's tightly wound set of loops of DNA. You need to unwind it before you can access a section either for replication or for transcribing a gene into mRNA. So let's kind of look at what this looks like. You have the chromosome, which is at the very bottom, and it's coiled up into chromatin loops. And those chromatin loops are wound on nucleosomes. And then you have DNA that's wound as well into a double helix. So it's actually really, really compressed. So when we actually look at a chromosome, like here, what we're looking for is a very tightly wound set of DNA. And chromosomes are paired, as we said. We have a diploid genome, which means that we have 2 copies of our genes. We get a gene from mom and a gene from dad. If it's the same version, same type of gene, it is known as homozygous. So if both chromosomes have the same type of gene, it's homozygous. If the gene is different, if you inherit one type of gene from your mom and another from your dad, that's known as heterozygous. And compound heterozygous if there's changes in the same gene at two different places. What do genes do? They code for amino acids. That's really their only purpose. They don't code for fatty acids, they don't code for carbohydrates. Genes are basically a sequence of 4 nucleotides, A, T, C, and G, and they code for 20 amino acids. So these 4 base nucleotides are the adenine, cytosine, guanine, and thymine. And they pair together. Adenine pairs with thymine, guanine pairs with cytosine. What happens if they don't quite code properly? Well, you can have a DNA-based mismatch. This is a fidelity issue. Now, what is a fidelity issue? If you remember back in the day when high fidelity from stereos was important, that is what you start with, what you originally started with, is what's being produced on the other end. There are several reasons for DNA damage. There's endogenous damage, and these are happening within the cell itself. One is from DNA-based mismatch. One is hydrolysis, and this is a reaction that's going to be mutagenic. It requires removal of both base pairs. You have to remove the one on the left and the one on the right. Oxidation, and this is a big one. This happens where? This happens in the nucleus, and this occurs from the mitochondria spilling over reactive oxygen species. Oxidized lipids and protein molecules can also manage to get into the nucleus and damage the DNA. Alkylation is another type that can cause issues with base pairing or can break both strands of the DNA. And it can be cytotoxic, which means the cell will die, or can be potentially mutagenic. There's exogenous damage. Now, this is stuff that comes from outside your body that can affect. UV radiation. This is ionizing. It is going to cause damage to your DNA. X-rays, cosmic rays, gamma rays are some others that can ionize and basically damage your DNA. Chemical agents and excessive oxidation such as ozone. Let's take a look at kind of what this looks like. There's base pair mismatches. There's single strand DNA breaks. There's adherence of one base pair to another. These need to be removed and replaced. And this is kind of the worst one, which is a double strand break. What does this all mean? When the DNA is unwound and you're making a messenger RNA, this happens through an RNA polymerase. You need to unwind the section of DNA, and you need to attach an RNA polymerase to it to begin transcribing each base. So you have floating around the base pair, the amino acid is attached to a triplet, a code triplet, that will be used to attach to create the sequence of mRNA. This happens with very good fidelity. The overall rate of transcription is one out of 10,000 to 1 out of a million. That's huge. In other words, it's very, very accurate. Nucleotides are added one at a time, a complementary DNA strand to form the mRNA. So these nucleotides are attached to a segment of RNA, and they fit into the polymerase and get attached. And this continues until the RNA polymerase encounters a stop sequence or stop codon, and then it releases the mRNA transcript. The RNA will travel out of the nucleus and get into the endoplasmic reticulum, and this is where it becomes translated into ribosomes. This is a huge and amazing mechanism of coding. And how all this works, to me, it's very amazing. So the ribosome will read a 3-base sequence, what's called a codon, and it will attach the right amino acid at the right spot. The translation rate is about 1 out of 10,000. Still fairly high fidelity, which means that out of 10,000 translation sequences, it'll make one mistake. Once it's complete, once the amino acid sequence is complete, it's going to fold in and become basically either a peptide or a protein. And this works kind of like a key in a lock. The shape of that protein or peptide is what determines its function. So let's look at the coding. Each set of three letter nucleotides codes for a specific amino acid, and you see there's some redundancy in here. And there are several stop codons as well. Now, this is the DNA part. Now, when you form the mRNA, the thymine is replaced with a uracil. So if you look at, it's going to be, every T is going to be replaced with a U. And the reason this happens is because your body doesn't want the RNA to be around very long. mRNA is much more easily broken down, and usually happens within 1 or 2 minutes if it has a uracil instead of a thymine. That's for safety reasons. So that's basically how genes, chromosomes, amino acids, mRNA, and DNA work. I hope this answers your questions, and I hope this was informative. Don't forget to like this video and subscribe.